for setting the stage for talking about connectivity and connectedness. This is, after all, the theme that unites us in different branches and disciplines of network science. And my own work is in neuroscience. And arguably, the brain is one of the most paradigmatically interconnected systems we can possibly think of. It is a complex network. Where's the trigger? Okay. It's a complex network par excellence, and we've been studying it in neuroscience for many decades now. And I think we're in the middle of a paradigm shift right now, where powerful approaches from network science and data analysis are teaming up with new acquisition methods and techniques for observing the brain. And I think also ultimately will link up with the kinds of approaches that Bernice is talking about from the social sciences. I'm going to talk about that in just a few moments. So there's obviously an enormous complexity that confronts us when we're dealing with the brain. There's networks on multiple scales from individual neurons. In fact, there's networks even within neurons. There's networks of individual neurons and brain regions that are interconnected. We can study those with different methodologies, optical imaging, histology, brain imaging at different scales. And then there's also different modalities of networks, anatomical relationships between regions or neurons, or dynamic relationships that are expressed in fluctuations of statistical dependencies, cross correlations, dynamic couplings that we think are really important for behavior and cognition, are really driving those important emergent properties of the brain. So there's networks across scales, across different modalities, and we're just beginning to get our hands around this subject by combining powerful new acquisition methods, neurotechnologies with data analysis and network science approaches. We're studying in my lab actually all kinds of different brains, including humans, obviously very important for us, but also animal models. Nematodes, fruit fly, the mouse brain, the rat brain, and the non-human primate. I'm going to focus here on humans, though, because that's where I think we're mostly interested in today. When we construct brain networks, it's actually not a small job. It requires a lot of technology, a lot of money, quite frankly, to pull in those data and then to process them into network form. And there's two different types of networks we typically work with in our field. On the one side, we're looking at relationships that are structural, wiring patterns, literally physical wiring of who connects to whom, or what brain region connects to what brain region in the brain. And that is shown here on the left with structural connectivity and structural networks that you'll see back here. For that, we have to process brain data into parcels or brain regions, estimate how strongly they're connected amongst each other, and then we make basically just a large table of what connects to what, which is a matrix, which is ultimately going to be our structural brain network. We also can record from brains as they're becoming active and dynamically engaged with their environment. We can do this with EEG, MEG, fMRI. These are all techniques that perhaps you've heard of. They're often in the media these days. And that gives us time series of observations of how neurons behave. And we can construct networks from those time series, which we call in our language actually typically functional networks or dynamic networks. And those are shown here on the right. Once we have structural networks coming from the anatomy, functional networks coming from when the brain is actually active, we can now interrelate these two major domains of how brains are structured and how they function. And network science approaches are extremely important for analyzing and understanding the organization of these brain networks. Structural brain networks that incorporate the entire brain of an organism we call the connectome, which is a term that we've coined here at IU actually 10 years ago now. And the field of connectomics has really taken off like a rocket. It's still doubling every year, I think, in terms of the number of publications and so forth that it actually attracts. So this is sort of the way that we extract brain networks from data sets coming from, in this case, the human brain, but also other species. My own explorations of this field began a few years ago in the deep, dark, early stages of connectomics in 2007, 2008, working with Patrick Hagman, who had developed some of the same approaches and ideas at about the same time. We got together back then and imaged a few individuals, a few people, using techniques that Patrick had developed called diffusion spectrum imaging, which allows us to map structural brain networks. And essentially, what we're getting is networks of how brain regions connect to each other in the human brain. That's essentially the output of that activity. We imaged at that time 5 individuals, and the first maps that we got looked sort of like this. You've seen this before in a couple of slides earlier. Essentially, it's a matrix of connections that say for each element of the matrix who connects to whom and how strong is that. Now, since then, 2008, this activity of mapping brain networks in this manner has taken off like a rocket, and there have been thousands of additional participants that have been imaged all across the world, and we have now access to literally thousands of individual data sets from different participant groups across ages, across clinical conditions, in all kinds of different variations. We can study individual people's network maps in connection to their behavioral, cognitive performance or other factors that we might be interested in. Yep. Right, so what I'm showing here is actually the structural connectivity, which we believe on time scales of seconds to minutes is relatively stable and unchanging. It will change over the lifespan. We've just begun to study this with our collaborators in China and in New York. And it changes across development, obviously. But this is a snapshot of a structural network. On top of that is dynamics, which is what these neurons and brain regions are actually doing, what they're talking about amongst each other and firing neural impulses. That changes on a very fast time scale. To track that and to relate it to cognition and behavior is an ongoing challenge that I think we're all grappling with in the field at the moment. And I'll say a few more words about this later on. This is actually just a structural network right here. There are many properties that we can diagnose, that we can describe from such a structural network. They're kind of listed here. I won't go through them in detail. This is sort of from our original publication, 2008. Again, these studies have been replicated probably hundreds of times at this point in dozens of different places around the world. Also across species, remarkably, many of these key features of human brain networks are also shared by other very different animals in many cases. We find modules and hubs and a structural core even in species like mouse and rat and even the fruit fly. There's sort of some general principles at work, we think, about how brains are organized that we can find across multiple different nervous systems. One of the properties that we were very interested in here, and we actually worked on in connection with our colleagues in informatics at the time, was to identify what I call the structural core or the rich club of interconnected brain regions. What is that? A rich club is a feature where highly connected parts of the brain, of which we have a few in our heads, are also highly connected amongst each other. They're forming, not only are they rich in terms of connections, but they're also forming a club. They talk to each other a lot more than expected by chance. This is a mode of organization that is sometimes seen in social networks, hence the name. And it was actually described and discovered by Vittoria Kolitsa, who was working in the School of Informatics a few years ago. And we, just a couple of years ago, 4 years ago now, started to become interested in this because there are theories about how the brain works that involve a central mental workspace, a core network where information comes together and is integrated. And we wanted to test whether we found a rich club of interconnected brain regions also in the human brain. We did that with Martin van den Heuvel, a collaborator from the Netherlands, who carried out the first study here. We did the analysis here at IU, imaging a number of participants. And to make a long story short, yes, we find parts of the brain that are not only highly connected, have many connections, and by that descriptor are considered to be hubs, but these hubs are also connected amongst each other, and quite often over long distances. Long-distance projections in the brain quite often tend to connect highly connected brain regions with each other. We call these informally the highways of the brain, the long-distance projections that carry presumably important message traffic because they are shuttling impulses between parts of the brain that are highly connected to start with. We found, if you're anatomically interested, where these are, and they check a bunch of boxes for people who are interested in brain regions and how they participate in cognition, and did a number of other analyses. Rich club organization was one feature that we've been chipping away at for a few years now. Others have around the world now as well. We're finding it very consistently, not just in human brains, but also, as I mentioned, in creatures like the fruit fly or even nematodes, so it seems to be something that is common to many different nervous systems. And underscoring once again the collaborative aspect, we worked out, we didn't work out the method to actually do this. It was worked out here at IU by our colleagues in network science and the School of Informatics. We hope certainly going forward to be working with them a lot more closely than in the past. And here's another set of connections that I wanted to make, and this goes back to Bernice's talk. The connectome kind of resides in this intermediate sphere here between it's a way of describing brain organization that is sort of interspersed between the very micro level of individual neurons or molecules, even genetic regulatory mechanisms, gene expression patterns, etc., which we think are very important for disease mechanisms, and above it, the sphere of behavior and social interaction. And this picture here, which I made a few years ago, resembles a lot what Bernice was talking about with respect to the social symbiome. In fact, this is, I think, a connection that we want to build on in the future as well. How are these different networks at different levels, how are they connected? We have social networks up here. We have brain networks in between. They have multiple scales, nested scales as well, from neurons all the way to brain-wide extended networks. And then we have below that networks within cells, molecular interactions, regulatory interactions that drive gene expression patterns, for example. We know that there are arrows between these levels in the upward direction. We know that gene expression patterns are very important for how the nervous system is built. The way the nervous system is built and functions is important for social interactions and behavior. But there's also arrows going the other direction. There's arrows that go from the social domain all the way down to the molecular. There's burgeoning literature now on how different patterns of social behavior, cultural practices can impact gene expression patterns, for example, and thereby modulate risk for developing certain conditions. We're very interested in these nested networks within networks types questions, and that's one thing that I see going forward as a real strong area of collaboration. Now I'm going to switch tacks a little bit and talk about the computational part of our work. We're not only looking at human brain networks and try to describe them using network science methods, we also try to build models of the human brain in computers that we can use to predict something about how brains behave, how they function. And I'm sort of repeating myself here. Here's the structural network at the top, a matrix of what's connected to what in the human brain. Below that is that summary diagram of who is interacting with whom in the brain, what brain regions are strongly coupled and perhaps also then strongly interactive dynamically. And that's a cross-correlation matrix in this case of time series that have been recorded from the same set of brains. We can now ask the question, how much of the dynamic interactions that you see at the bottom can be predicted on the basis of the pattern of what's connected to what? After all, knowing what's connected to what is a nice exercise, but ultimately fruitless if that's all it is. We also want to know how it actually underpins the function of the brain. And we can ask questions about prediction. We can also ask the opposite question, which is if we know how dynamic brain regions are interacting, how brain regions dynamically interact, how much can we infer about the connectivity, the structural connectivity between them? That turns out to be a much more difficult problem, a so-called inverse problem. We can also use connectome data sets, like the thing that you see on the upper left, and we can put them into a computational model and build a computational model of how the brain behaves dynamically. And we've done that over the years, I think, quite successfully. Here's how we've started doing this. This is a matrix of anatomical connections coming from the macaque monkey, non-human primate, and it was assembled for us on the basis of hundreds of reports of what connects to what using histological studies in the macaque species. And this is literally several decades of work that have been assembled together into this one data matrix. And when we first started working on this in 2007, we had no human connectome data yet. This was just a gleam in someone's eye, my eye for the most part, that we might have that data one day. We had it the next year, which was quite unexpected. This is what we worked with in 2007. It's a matrix of what connects to what, and then we have biophysical equations that describe what neurons do dynamically. Conductance-based methods that say what neurons do electrically, so to speak. We combine these two, we create a dynamic simulation that can be passed through fMRI filters, very analogous to what happens when we put in a scanner, and we're getting back a correlation matrix of time series, which is our functional connectivity network. At the time, 2007, we had no idea what the real brain of the macaque looked like. We had no data about that. It turned out that we were actually quite good in our prediction. A couple of years later, Yasushi Miyashita from the University of Tokyo contacted me, wanted our simulation data because he had just acquired some monkey fMRI, which is very hard to do, as you can imagine. Monkeys don't speak English, so you can tell them to hold still, but they won't do it. He had a correlation matrix just like this, empirically obtained, and we compared side by side, and here's the comparison, somewhat colorful. On the left, the empirical correlation matrix, just one triangle of it because it's symmetric, and on the right, our model from a few years earlier, which is not perfect, as you can tell, but also is not totally imperfect, given that there was no model tweaking and tuning and parameter fitting and whatever going on here, and given that the data matrix we worked with probably wasn't perfect. So we're quite successful in taking anatomical data, structural data, computing something on these data, and then comparing it to how empirical data look like. Why is this important? We can apply this to the human brain. Everybody these days wants to build a model of the human brain in a computer, right? The Europeans have just committed, well, they may uncommit, but they've committed €1 billion to that over 10 years. And we actually can do this, not at the individual neuron level yet, because we don't have the data for that, but we can do it at the regional level quite well. We should have asked for $1 billion, actually, but that just occurs to me. We can actually do this quite well. We're tweaking our methods and are beginning to get better and better at it. Ultimately, what I see in the future is to build computational models that can be used as predictive tools for plotting therapeutic interventions, perhaps for modeling a patient's brain in detail, as an auxiliary tool for understanding that person's brain dynamics better, to make a prediction about when a cut is made here or tissue is removed or when there's a surgical intervention, how would these dynamics be impaired. We're also working on much simpler models, which I will just briefly flag here, that are not as computationally expensive as these very detailed biophysically based models I showed you earlier. These are models that are much more like social network models, and we're beginning to work now with our colleagues in informatics on making better models like this and actually using them on brain data. It's our first shot at this, which really I thought good given how simple this model actually was. On the left, once again, the left triangle of this matrix is a depiction of empirically recorded human brain dynamics in the so-called resting state, dynamic signatures of how brain regions are interacting. Underneath it all is the connectome, which drives this. We use the connectome to then plot likely paths of communication in the brain. The connectome is a wiring diagram. Very few brain regions are directly connected. To pass signals between them, quite often they have to go through intermediate steps. We're plotting all of these intermediate steps. We're trying to predict on the basis of how direct or indirect they are connected with each other, how strong would their functional coupling be? How likely would it be that they can actually communicate in real time? And that is, our prediction is shown on the right side of this square matrix, which is a prediction based on very simple physics-based communication models. These are so simple, these models, that are actually analytic. We can actually solve them in a computer in under a second. We get a very good prediction of what that brain's dynamics would look like, and a few colorful brain images here on the right to prove that point even more. Why is this interesting? Well, first of all, the fact that you can predict something about the human brain on the basis of an extremely simple model so accurately is remarkable, I think. Secondly, we can, because this works so fast and is so clean, for lack of a better word, in terms of the analytics, we can apply this on a grand scale. We've done millions of these simulations now, trying to find, for example, identify crucial parts of the brain that when they are impaired, will really impair functional connectivity and functional brain dynamics. The fingers are pointing all the way in the direction of hubs, highly connected. This is perhaps not surprising, highly connected brain regions, and people have now mapped these very in much detail. When they are impaired, that is when things start going wrong at the cognitive behavioral level, for example. So that's a very important direction for us in the future. I'll summarize here. So connectomics and network science approaches are beginning to tell us a lot about how the architecture of brain networks is organized. At this point, I always like to stress that it is very early in the game. We are less than 10 years into this. I remember the days when we had no data at all. Now we are drowning in data. The Human Connectome Project, which is very much analogous to the Human Genome Project in many ways, which was inaugurated by the NIH in 2009, is finishing next year with a data output that is close to 1 petabyte. You actually cannot move these data around on the web. You have to actually order the data on hard drives. It's the only way you can actually get the data, because there's no way to move a petabyte of data over the web. An enormous amount of data is coming our way, and I think connectomics, especially in connection with network science, where we have, I think, extraordinary strength here at IU, is going to be a very important part of human brain neuroscience in the future. What is a theme that we often come back to now in our own work, whether it's lesion mapping, whether it is looking at psychopathology and schizophrenia, looking at degenerative conditions, whether it is looking at individual differences among humans, looking at development, hub regions or hub nodes are of critical importance in the human brain. This is a major insight, I think, because even though we knew for many years now that some parts of the brain are more important than others, we didn't know why. Now I think we do know one of the underlying reasons for why they're so important is very simple, because they are highly connected. We can also look at their connections amongst each other, and that's the so-called Rich Club phenomenon. Very interesting idea, I think, that is starting to gather momentum also in the field. It's been studied in many, many places now. We find it across species. It's telling us something important. When there's something you find across species, from fruit fly all the way to humans, and it's held in common over these very different neural network organizations, there's two ways to think about it. It's either trivial, which I don't think it is, or it is actually telling you something very important about how brains are organized. In this case, the importance of integrating information in a common workspace. And so the topic of integration, connectedness, is one that overarches all of this. Network science is about relational data. It's about studying connectivity in its own right. And neuroscience, I still have sometimes to convince people that connectivity actually matters for how the brain works, but I think that cohort of people gets smaller and smaller every year. Connectivity is obviously important in the brain. It is, however, not the only thing we have to understand about how the brain works. There's networks underneath that involve tissue, metabolism, energy, gene expression patterns, etc. And there's networks that, of course, the brain engages in when it's becoming active in the behavioral and social and cognitive domains. And so for me, going forward, there's many challenges here, but one of my pet challenges is to try to connect networks across different levels. And for that, I think we have many opportunities with the colleagues we have here at IU. Thank you very much.
Architecture, we're really not looking at the neural behavior, we're just looking at how they're connected. So I wonder what the connection between all this architecture that you're finding here and all those results that we have done by looking at new places, well, you have to come to
yeah.
What's the connection between, because when you do that, you do the local field potential.
Yeah, so it's really a question about scales of organization, and there's a long tradition in neuroscience to look at individual neurons and what they do. And techniques to do that are very sensitive, but also very difficult to control. It's difficult to get access to all the neurons in a person's brain, for instance. In fact, impossible at this point. At the same time, when we do imaging, we only see cubic millimeter sized chunks of tissue which contain hundreds of thousands of neurons, and we can't see the individual neurons anymore, and we're getting a very sort of temporally blurred signal from that as well. So how do we link up these two things? This is really what the Brain Initiative is all about. The N in brain, and that acronym stands for neurotechnologies, and it's about finding new ways of recording from nerve tissue in model organisms, but also ultimately in humans, that can exactly address the question you're asking. How do we connect what we already know from electrophysiology, which are very sort of punctate observations, single events in a single neuron in connection to behavior, to these very large-scale observations that are, however, quite spatially and temporally blurred. Somewhere in between is the sweet spot, and I think the Brain Initiative tries to drive research in the direction of identifying where that is, and we're now getting, for the first time last year, zebrafish. It's very small, translucent. When it's a larva, it's translucent animal. Zebrafish recordings, optical recordings that are actually whole brain, individual neurons. We get all the time series, and guess what? People are beginning to do exactly the kinds of analyses that I've been talking about here. Network analyses, dimension reduction techniques, functional connectivity. These techniques that we've developed a little bit on the imaging side are being imported now into studies of much smaller organisms where we have complete access, however, to what individual neurons are doing and how they evolve through time. So I see a very interesting, challenging future ahead for us. Yeah.
Do you see the error between the predicted networks and the empirically observed networks as resolvable, or is that fundamentally inherent in that regard?
That's an excellent question. Some of the mismatch between our models and the empirical data are probably because the data going into the models isn't perfect to start with. So our estimates of structural networks, for instance, our connectome mapping efforts are still riddled with all types of errors. We see connections sometimes where we know that they couldn't possibly be, or we have connections that we know are there, but we can't see them. So there's all different types of errors involved in that. So we think that if we get better connectome mapping techniques, our data gets more accurate, our models will get more accurate as well. But there's also an element of variance that can't be explained, I think, by structural connectivity alone. There's physiological mechanisms as well that operate at sort of a circuit or neural population level that we have to incorporate. So our models have to get better. For me, the glass is half full. If we can get a very simple, extraordinarily simple, abstract way of computing network statistics, and we can match that up with functional connectivity data coming from live beings, okay, in an MRI scanner, that tells you something important, I think. That tells you that these models that are very simple, nevertheless are tapping into something fundamental and are telling us something how the system actually works. That's my hope for the future also, is that models that we can use in our work are going to be quite simple and powerful. If we have to build a human computer and a human brain in a computer cell by cell, that's not a future I want to be a part of, okay? Because then all we do is we replicate the system, and we still don't understand it, okay? And this is sort of where I think some very high-profile brain projects in the world are kind of foundering a little bit. Understanding does not necessarily come from replication. We have to find the principles, the computational principles underneath that are driving how the brain is operating. We're trying to do this, and we're trying to use network science methods for finding that out. Thank you.